Okay, record on this computer. Okay, here we go. So today we're going to talk about scrub typhus, and our discussions are still within the family Anaplasmataceae. Yeah, I don't know how to spell it. Anaplasmataceae. Well, let's review from your guys' knowledge what's in this family and what's unique about this family. For those in person and online, what's in this family? Like, what organisms are in this family? Okay, Wolbachia. Something was written in the chat. Orientia, Rickettsia. I'll draw these trees. I'll try to draw them somewhat. Orientia is close to Rickettsia. What else is in this tree? We should know from the tick lectures in the chat. Yes, I'll get to that in 1 second. I'm still looking for like 2 more big ones from the tick lecture. Yep, Ehrlichia. I'll spell this wrong too. Hopefully not. Ehrlichia and Anaplasma. We should know about these. And we should know that this family, like, this will be on the test for sure, because this family is so sort of like intricately linked to vector biology. And some interesting more things about this family, which you should remember, is I kind of talked about how this family is kind of thought to be sort of like share a common ancestor with mitochondria. So if you're into like the evolution of mitochondria, the evolution of eukaryotic cells, it's thought that some common ancestor of these creatures was kind of like imbibed by, and that created the first eukaryote. So these are very small genome, obligate intracellular parasites. So we want to kind of like know these characteristics. And these are gram-negative or gram-positive. Do we know what that is? Lauren? Gram-negative or gram-positive?
I don't know what they are. Like, I know what the difference is.
What? Okay, so help me out. What's gram-negative?
Gram-negative is like thinner cell walls.
Thin cell wall and 2 membranes. So there's an inner and an outer membrane, and there's kind of like a thin cell wall between in gram-negative. Gram-positive is different. How? A giant thick cell wall and just 1 inner membrane. And this family, these are gram-negative. So they look like this, their outer membrane. These are important things to remember about this family of bacteria. And many of these Wolbachia, what's unique about them, they're non-pathogenic and they manipulate insect reproduction. We know this, but this is a good review. These things, Ehrlichia, Anaplasma, and Rickettsia, cause diseases, and oftentimes they're spread by ticks. Rickettsia is spread by ticks, but also by fleas and lice, which we'll learn about more later. And Orientia is the one we're going to talk about today. So Orientia is the special person we're going to talk about today. Not person, but bacteria. And Orientia causes a disease which we call scrub typhus. And the vector is mites. Specifically, mites that look like this. These are what we call chiggers. And we'll talk about them a little bit more. But first, let's go back to scrub typhus. So the species is Orientia tsugamushi. This name is Japanese. This disease was described by the Japanese. And this is what I looked up. Maybe somebody should fact check me on this, but supposedly Tsutsugamushi, when I looked it up, it translates to something like small and dangerous. which would make sense because it's spread by these mites that you can't see. But I'm not a Japanese expert, obviously, but that's what I looked up. And it's called scrub typhus because you get it in the scrub. That was like the common name. You get it in the scrub. What is the scrub? It's like the same thing as kind of like the bush, like in the woods, in the jungle. Okay. And we sort of like had no experience in term when I say we, I mean like United States, like Western Europeans really didn't have any experience with this disease before until World War II. So in World War II, there was the Pacific Theater, and so half of the entire World War II was involved in essentially like fighting on islands in the South Pacific. And many of these islands were owned and operated by the Japanese Empire at the time. And so when we were fighting in World War II, that's when we first started getting exposed to this disease, and they started calling it scrub typhus. Okay, so more on the history of it. There's some interesting facts. So in the Pacific Theater, 35% mortality of the troops. 35% mortality of infected troops. So if you were a troop fighting in World War II in the Pacific Theater and you got this disease, 35% of those people died. And this is actually like a famous theme in vector biology. You see it recur all the time where these wars are fought. And when wars are fought, epidemics break out. Why would that happen? From the patterns that I've talked about in the past, why would epidemics happen to break out during wars? Yes, there's mass quantities of people moving into new areas in which they've never been exposed to before. They don't have any immunity against the diseases, and they're also moving around and being exposed to new vectors, and potentially they're carrying stuff. And also, yes, in the chat it says hiding in the bushes. Yes, exactly. Like that's how they got this disease. All of a sudden there's a bunch of troops hiding in the bushes. And actually in the Pacific Theater, you were more likely to die from scrub typhus than you were to be shot by a bullet from the enemy. So when you fought in World War II, you were literally more likely to die from this disease than you were from being shot by the enemy. And that's also like a pattern that you see in other wars. Many times what kills the troops in high quantities is outbreak of disease and not necessarily what we would call the enemy. So this is, I don't know if this poster dates to World War II, but this is a poster that comes up when you Google scrub typhus. It's interesting to look through the techniques that they proposed would help them. They say always apply insect repellent, always wear clothing impregnated with insect repellent. I can't remember what the chemical is they put in there, but the Army specifically like impregnates their clothing with a thing that kills arthropods. I'm trying to remember. Do you know Ifan, do you know the chemical? Permethrin. It's permethrin. Permethrin. They put permethrin in their clothes to kill things that like this that would spread diseases. Always keep sleeves rolled down, so which is hard if you're in a tropical environment. Very difficult to do. Avoid sitting down or lying in the damp grass. Also very hard, as Lewis mentioned in the chat. If you're trying to avoid enemies shooting at you with bullets, probably hard to avoid lying down in the damp grass. Scrub entire body with soap. The most effective way that they were able to sort of like get rid of scrub typhus in regions that they were working with is they would literally just like burn all the vegetation, burn it down and clear an area. And then if you cleared the area, okay, then the scrub typhus would go away. But the scrub typhus deaths outnumbered deaths killed in action in the Pacific Theater. So it's a very interesting disease. And this is one reason why actually the Army still has a very vested interest in researching vector-borne diseases. So many of the advances in vector biology are funded and driven by interest from the Army. That's a pattern that recurs today. Okay. So back to the slides. I don't know. That might be true. The comment is that CDC was potentially established because there was malaria amongst the troops. That would make sense. It would not surprise me at all. I don't know if it's. I don't know. I don't know if it's true or not, but I'm assuming you're right. That would make sense. This is what they call the scrub typhus triangle, the Tsutsugamushi triangle. This is kind of like the region of the world that's endemic with Orientia, with scrub typhus and the mites. So obviously it includes the South Pacific, New Guinea, and many of the regions, obviously, where World War II was fought. Japan, the Koreas, tell me if I'm wrong, but China, Iran, and now it's sort of like expanding. You see these things pop up in different regions, so even outside of this quote unquote triangle. Australia, what is New Zealand over here? So maybe New Zealand's not in the triangle. But this is kind of like the region of the world where you find this disease. And the other interesting thing is this is a serious disease that almost like nobody has heard of this disease. Almost 1 million cases a year with 10% mortality. And this is actually more deaths than dengue fever. So we care a lot about dengue. And I fact checked this. So I looked on Google how many deaths occur from dengue every year, 14,000 to 20,000. So that fact check works out, because if there's 1 million cases a year with 10% mortality, that's 100,000 deaths a year. That's a lot of people dying from, especially from a bacterial disease that can be treated with antibiotics. So many cases, the people who are dying of this are probably poor people who are in the bush. They get a disease, they don't quite know what it is, they don't go to the doctor, and then all of a sudden it gets really bad and they die. And it's probably at that point too late to go to the doctor and get antibiotics. So it is a very serious disease, and I guess we would call this something we would call a neglected tropical disease for sure. Kills more people than dengue. So it's fascinating that there's a lot of interest in dengue, but not so much interest in research. You don't hear about it. You never heard about it. Yeah, see, that's the thing. It's strange. I had never heard about it either until I started studying vector biology, but it kills a lot of people. I think one reason you don't hear about it is because it's insanely difficult to study. The reason it's insanely difficult to study is because, well, I'll talk about it in a little bit, but because it involves mites, which are extremely difficult to culture. It's a very dangerous pathogen, so it's hard to get the infrastructure to actually study it. But we'll talk about that in a sec. Okay, let's talk about that now. So it's BSL-3. This should be review now. What's BSL stand for? Biosafety level. So you need special facilities to study this. You can't just study it like in your lab. It's very dangerous. And not only is it BSL-3, so again, this sort of points to like, why is this disease difficult to study? Probably why do we not know much about it? One reason is because it's BSL-3. A lot of people don't want to work with BSL-3 organisms. You also need a lot of funding, you need a lot of infrastructure, you need a lot of money. Second thing is it's obligate intracellular. So it's not like you can just readily culture the bacteria. It's very difficult to culture the bacteria. You have to culture the bacteria in theory, what would be in cell culture. Cell culture is pretty expensive. So it can be done, but most of the people studying this culture Orientia in human cell culture or monkey cell culture, I think. There isn't I'm not aware of a mite cell culture. So that's essentially the other half of the disease transmission equation essentially can't be studied. Or if you want to study it, the actual transmission of the disease, you have to culture the mites. Why are the mites difficult to culture? Why is it hard to grow mites in the lab? Or is it easy? Is it easy to grow mites in the lab?
Aren't they really small?
They're small. That definitely makes it difficult. But I mean, we have microscopes. That's not the main limitation. It's about a year. Yeah, their life cycle is about a year. I think it says 2 to 12 months later. These aren't the main reasons. Why else is it difficult to culture these mites? What do they eat? What? Actually, they don't eat blood. They're not hematophagous, but they're biting animals. They do need to. They're essentially what they do. Look in the chat. They eat flesh. Yes, that's essentially true. They they have a vast taste for flesh. What they do is they spit on your skin and they dissolve your skin and they feast on like your skin cell soup. So they don't suck blood. They don't really bite you. What they do is they sort of like regurgitate dissolving fluids onto your skin and then they suck up the skin Slurpee. But it's difficult to culture them because so then how would you feed them? How would you feed the larvae? You have to feed them on vertebrates. So you have to give them some source of like their food, their skin food. And what do we know about the difficulty of vertebrates? Why is it hard to work with vertebrates in some sense? Yeah, there's IACUC. There's IACUC protocols, which are essentially like, these are the regulations under which you can work with vertebrates and you have to follow these special rules and there's special ethical rules and it's expensive, it's not cheap. And so essentially to work on Orientia, you have to grow the mites, and to grow the mites, you have to have vertebrate approval, and all this sort of infrastructure of this research is in the context of a BSL-3 facility. So this is a very, very difficult system to work with, and which is probably why we know almost nothing about the disease and why nobody really hears about it. It's very difficult to work on. Okay, little quick review. Back to this grouping. What's unique about Orientia and Rickettsia in comparison to Anaplasma, Ehrlichia, and Wolbachia? So what's unique about this group compared to this group? Where they live? What's different? Yeah, these ones live inside of vacuole, and Orientia and Rickettsia live directly in the cytoplasm. So Orientia is one of these obligate intracellulars that lives in the cytoplasm. That's another good point worth pointing out. Okay. We did that. Okay, let's look at the mites. The mite phylogeny. So here's a picture of the mites. The family, which you want to know the name Trombiculidae, trombiculids. So if I say trombiculids, these are what we call the chiggers. These are the family of mites that spread this disease. And the genus that spreads them is Leptotrombidium. And I probably will ask you this, so I do kind of want you to know this, that Leptotrombidium mites are the chiggers that spread this disease. Of course, these mites are where? In this big tree. Where are mites in this vast arrangement classification of organisms? Chelicerates, so they're in here. Within here, where are they? Where? It's on the picture. No, the larvae only have 6 legs. We'll get to that. Looking in the chat. They're definitely chelicerates. Yes, that's correct. They fall under acari. So acari includes both the mites and the ticks. Okay, so they're very, very closely related to ticks. I made a comment in the tick lecture that ticks are essentially like large mites. They fall in acari with the ticks. Okay, just to kind of like show you that, again, there are other mites that are medically important. So these are the chiggers. These are what they look like. This is the larval stage, because the larval stage has 1, 2, 3, 4, 5, 6 legs. These mites are scabies. These mites are what dust mites look like. So there's other mites that are medically important. Scabies will like parasitize you. Dust mites will cause allergic reactions. But these are the ones that vector diseases like scrub typhus. I've showed this picture before, but now it's worth bringing up in the context of this disease. What does this morphology tell us about how they eat? This is their mouth. Yeah, they're like chewing and sucking. So this is not a selenophage. This is not something that's going to stick its mouth parts inside of you. This is not like a tick. Here's a tick mouth part. It's not like a tick that's going to stab it inside of you. It's not like a telmophage, which is going to like lacerate your skin. There's no sharp blades. This is just going to suck up stuff, kiss you with its lips, and eat your skin cells. We've seen this picture before. This is what I was describing, this sort of like wound in the skin. It's called the stylostome. So they're spitting these digestive juices. Their digestive juices in their saliva cause like digestion of your skin, like a hole in your skin, and they just sort of like suck up. And this is the wound. Okay. So if that's the wound, how do you get the disease? How do you get, if the bacteria, if the Orientia are in this creature, how do you get the disease? How do you get scrub typhus from this thing? Yeah, because this is not accessing your bloodstream. So in theory, the way that you get the disease is you smash this thing, you swat it, or you scratch it. You smash it. Now you've all of a sudden you release the bacteria, and maybe you'll scratch the bacteria into your skin. And if you do that, you get things that look like this. And I would imagine these are very itchy. So you can imagine when you get a mosquito bite, you sometimes will itch it till it bleeds. And then you get a nasty infection. So the other thing is keep calm, don't scratch, don't itch. And if you get scrub typhus, you'll get what looks like this. This is a special term called an eschar. An eschar is a scab that's black, a black scab. And this is a very, very famous symptom of rickettsial diseases, of scrub typhus. If you get this black scab, that's a bad sign. It's a bad sign that you might have just infected yourself. So let's look at the life cycle of the nymphs. Which is the stage that transmits the disease? The adults, the nymphs, the eggs, the larvae. What? The larvae. The larvae are the stage that transmit the disease. So as the larvae come out, these things look for, I wouldn't call it a blood meal, but like a skin meal. They look for like a skin meal. How is this different from other vector-borne diseases that we've discussed? This one is unique. How is it unique? If a tick comes out of the egg and bites you, are you capable of getting a disease? What? No, not really. I mean, unless it was like maternally transmitted. But if a tick freshly comes out and it's a clean tick and it bites you and it sucks your blood, there's no way you're going to get a disease because that tick has not picked up any pathogens yet. Most vector biology is like the mosquito or the tick, where it bites a source, a reservoir, picks up the disease, and then bites again, a second blood meal, and then gives you the disease in the second blood meal. This is unique. The chiggers bite you right after they come out of the egg as larvae. They're preloaded with the bacteria. What does that mean about, why does that mean about the bacteria? It's maternally transmitted. That's a very important thing. So So actually, if we look at this figure, if this is truly from World War II, in World War II, they had this completely wrong. They thought that rats were like this reservoir, and then chiggers bite the rat, and then the diseases spread from rats to man, in a sense that like they thought it was like a zoonosis. That's not what happens according to our understanding today. According to our understanding today, the Orientia are already in the mites, and they're maternally transmitted. So this is actually kind of scary, because what this means is, let's say this is an adult mite, 8 legs. It's got the bacteria. It means that when she lays eggs, every single one of these eggs is endemic for Orientia. This is unique, and this is new compared to everything else that we've discussed before. Everything else we've discussed before is in a reservoir, it gets picked up in a zoonotic cycle. This is not that. It's endemic in the mites, passed on maternally in the mites, and then when they come out of the egg and they bite you, you get it immediately. Also, the life stages, the other life stages of the mite, if my understanding is correct, they do not blood feed. They walk around in the dirt and eat like insect eggs and things like that. They're kind of like predatory mites. So the other life stages don't blood feed. It's only the larvae. It's only the larvae that we worry about here. Other points worth mentioning are that the life cycle is 2 to 12 months. So again, this is not a fast organism to grow in the lab. Another reason why it's difficult. And it is small, and that does contribute to why it's difficult to work with in the lab. So let's ask a question, sort of like a more creative thinking question, which should be able to answer. What's the reservoir for the disease? What's the reservoir? The mite. In the chat, somebody, Lewis said the mite. That's correct. The mite itself is the reservoir. The mite itself is endemic. Okay. Usually how they detect this, it's expensive to diagnose, in a sense that you could either do PCR, so you could PCR to see if somebody had it, or check for antibodies. But most of the time, I think when they try to detect this, they're trying to detect with PCR. And again, it's not a common disease. So one of the issues with many of these diseases is, so let's compare it to dengue again. Let's compare it to dengue. In dengue, there's 390 million new infections annually. Okay, so 390 that's 390,000, 390 million dengue cases. Let's go back to that one slide and let's look at how many. 1 million. It's only 1 million cases of scrub typhus. So in terms of like infrastructure of dealing with disease, like our public health infrastructure of dealing with diseases, which one is our infrastructure probably more equipped to deal with? Like which one are our doctors going to be more familiar with? Dengue. They're going to be way more familiar with dengue. They're going to be familiar with how to treat dengue. They're going to be familiar with like they're going to have all kinds of different technologies to detect dengue. They're in theory, they might have different treatments for dengue. So like the issue with these diseases, which we call the neglected tropical diseases, is they may be very serious. They may be very serious diseases, but the rate of incidence is so low that what are the odds that the average doctor has ever even seen a case of scrub typhus? It's probably very low. So when a doctor sort of like encounters this, many times they don't know what it is. They don't know what to do about it. They don't know, they've never seen it before. So these diseases are often tough to deal with, and often it makes, well, the point here is that it makes detecting them expensive, because what pharmaceutical company is going to develop a test that they can sell to test for scrub typhus when this is essentially the product market compared to the product market of dengue? Like, there's not going to be an infrastructure of sort of like treatments and ways to deal with these diseases. So if there's no way to like sort of like automate the detection, or like have like a real quick way, a test that a company is producing, usually like the go-to thing that people do is like PCR. And again, like if you go to a hospital, is there a person there that can do like a PCR from blood samples to detect for Orientia? The answer, you might think yes, but the answer is actually probably no. There probably is not some like random person there that can just like do a PCR on blood analysis. So these are going to have to go to special labs, and it's going to be, in a sense, tough to detect, but in another sense, easy to detect if you just know how to do PCR. But sort of the main point is that the main infrastructure to deal with this type of a disease is not there. And that's in part probably what contributes to its mortality. Is there anything that's worth mentioning from the end of this slide? This is a really interesting slide that's worth pointing out. So this comes from a paper here I'm citing. And this is a pattern that we've seen with filarial worms. So the case with filarial worms was, what was unique about the behavior of filarial worms when they're in the blood? They would only come out at night. They were nocturnal. And the reason they were nocturnal is because they wanted to coordinate the point at which they were picked up with the point at which mosquitoes feed. They want to complete that life cycle, and they need to get into the mosquito. So they would become active when the mosquito was feeding. Similar thing kind of occurs with Orientia. So Orientia replication, so like when the Orientia is dividing and like growing and magnifying the quantity of its infection, it's coordinated with the host life stage. So in the larvae, here's number of Orientia. So this is number of Orientia on the y-axis. X-axis is different stages, okay? Which stage has the higher numbers of Orientia? The larvae. And which stage spreads the disease? The larvae. So the Orientia replication of the bacteria, it somehow knows when it's in here, and it knows when it's in the larvae, I better start replicating because I want to get transmitted. It's interesting. And in the nymph and the adult, it's not replicating as much. This is interesting. I don't know how to answer this, but a question just came to my mind. You would also think that it would want to replicate a lot in the adult. Why? Yeah, it wants to get into the eggs. So maybe perhaps this study should go into a higher fidelity, a higher resolution, and check like, well, where is it? In the adult? Is it in the ovaries and the testes? Is it replicating a lot in the ovaries and the testes? And perhaps in the larvae, it's replicating a lot, maybe like in other regions of the body. That would be an interesting study to do. I don't quite know how to answer that question. But again, we don't know so much about this disease. Do you guys have any questions? That's kind of the gist. This is a shorter lecture because, again, we don't really know very much about this.
So the meaning of so I knew mushi dog because Japanese is my second language.
Okay, so you're saying mushi
Mushi
means
dog.
bug.
Yes.
So that's probably what I thought, like little thing.
Yes, and then Sika I googled and
So it's bug illness.
illness bug.
Illness bug.
Yes.
Awesome fact checking. Good. That makes sense. Okay. So they must have known like long ago. They must have known that the disease comes from these mites. Like they must have figured that out. It's interesting to see like how much people kind of like figure out even in long ages long ago. Okay, that's it. Good lecture. Everybody have a good weekend.